All right. Good morning, everyone. We are recording now. I do have a couple of really important announcements. Obviously, we're running a little slower than what's in your syllabus. So I've already postponed the exam for Section 1. For you all, it's going to be postponed until Thursday. Okay? So you have about 10 days. That's going to be February 19th, correct? February 19th. Sorry, 20th. It's Thursday. Is it the 20th? Okay. And then the first section is postponed until that Friday, the 21st. Okay? All right. So that is I'll make a formal announcement today after class, and just make sure that you put that on your calendars now. So we actually have 3 class periods before exam 1. Yes?
So in the book, sometimes things go into some more specific detail than what we go over in class.
Right.
Should we expect some of that specific detail in the exam as well?
Right. No. Great question. The question was, sometimes the textbook goes into a lot more detail than what we do in class. I'm only going to test you on what we talk about in class. So there's not going to be any surprises. Yeah, the textbook does go into a little bit more detail, and that's why I have you buy it. It will help with a more sophisticated understanding. But really, like I said, the test is difficult enough just by what we talk about in class. So thank you for that question. Any other questions? All right, well, let's go ahead and get started. Today, I hope to bring a lot of things together, right? We've been learning a lot of different concepts in a very compartmentalized way. So I really want to bring it home today. Any questions about what we discussed last time? Do you feel pretty comfortable about G-protein coupled receptors? At this point in your academic career, you may be seeing it here in physiology, in microbiology, in biochemistry. Hopefully it's really overlapping at this point. I do want you to memorize the pathway, but again, remember this is a multiple choice exam. Okay, so you just have to recognize it, know the order, and just recognize it. You're not going to have to, like it's not an essay, it's not short answer. You're not going to have to write out phosphatidylinositol 4,5 bisphosphate, right? On the exam, it'll say that, but then in parentheses, it'll say HIP2. So you're not going to have to memorize those names, okay? Any questions about what we discussed last time? All right, so I better stop for a minute. Okay, so let's go ahead and get out your top hat devices, everybody. Let's go ahead and start again with top hat. All right. Let's see. Okay. Steeping some tea. All right. All right, so here's the first question for today. We talked about this last time, pretty easy one, I think. True or false? Some receptors act as a ligand-gated ion channel. Is this true or false? Is this true or false? And you can actually turn to your neighbors, you can talk about it if you're not sure. I'm going to roll out a couple of new terms with this question. All right. All right, we have about 43 students, 44 that have answered already. Give you about 10 more seconds. All right, does anyone need a little more time in this room? Okay, yep, I can give you a few more seconds, no worries. Just give me a thumbs up when you're done. I still have one more student that's... Pretty good? Still working, still thinking. Okay, nice. Anyone else? You're still... Okay, did you get it? Okay, close. All right. We will have another question too, so if you don't get it on this one. All right, so I'm going to go ahead and close this one, and of course the right answer is true. Okay, great job. I'm not gonna go over this again, but I do want to give you a term associated with this. Okay, so ligand-gated receptors that are ion channels are called ionotropic. Ionotropic receptors. Ionotropic receptors. Can everybody see that all right? Ionotropic receptors. Ion kind of gives you an idea of what these receptors do. When the ligand binds to these receptors, they are ion channels, so they open up, allowing for electrolytes to move across the membrane. All right? Now, G protein coupled receptors are called metabotropic. Metabotropic. So just a couple of new terms. Just want to make sure that you have those terms in your glossary. Ionotropic receptors are receptors that are ion channels. Metabotropic receptors are G protein coupled. All right. All right, so how about this one? Which G protein coupled receptor increases the level of calcium within the cell? Is this GS, GI, or GQ? GS, GI, or GQ? So these are the type of questions. I'll also give you another type of question in just a second, what you can expect on the exam. All right. Give you about 10 more seconds. We're up to 49. All right, does anyone in this room now need a little more time? Pretty good? All right, so close this one up, and the correct answer, of course, is C. Yes, good job. About 75% of you got that right. Most of you did. GS and GI, they alter the levels of cyclic AMP. Okay, GQ is actually the one that causes an increase in intracellular calcium. All right, so the type of questions you might get on the exam, I get this question a lot. Let's just say we're talking about the GQ coupled receptor. What if I add a drug? These are more application type problems. What if I add a drug that's going to activate PLC beta, right? So now in your mind, you should automatically think, okay, that's the GQ coupled pathway. What's going to happen to calcium? Calcium's going to rise, right? If you activate PLC, calcium's going to rise within the intracellular space. What happens to G alpha? If I have a drug that just activates PLC, what's going to happen to my G protein alpha subunit? What do you think? It's the G alpha, GQ alpha specific. Do you think this drug is going to have anything to do with G alpha?
So it goes down.
Yeah, it's kind of a mind bender in some ways. If you activate downstream in the pathway, nothing happens to the G proteins, right? So those are the things that I want you to think about too. What is upstream from that enzyme? What is downstream from that enzyme? So calcium should go up, DAG should go up, IP3 levels will go up. Nothing should affect, right? That's not going to affect the G proteins at all. Okay, does that make sense? Okay, so those are the type of questions I want you to expect on the exam. It's not going to be drawing it out. It's not short answer. It's going to be these kind of application type problems. Okay? All right, great job. Okay, so that's it for today on the top hat. Let's go ahead and continue on. Last time we did start with cell volume regulation. This is actually good cell physiology. All right? G protein coupled receptors, you might see that in cell biology, I would say biochemistry, right? These are relevant to a lot of different disciplines, but what I'm going to talk about today is very specific to physiology, okay? So here's where we left off. We were talking about cell volume increase. It's called regulatory volume increase and regulatory volume decrease, okay? So if you actually drop a cell in a hypotonic solution, immediately the cells are going to swell, okay? The cells actually have mechanisms in place to try to shrink their volume in the face of a hypotonic solution to maintain cell volume in a homeostatic range, okay? So there's mechanisms in place to try to keep it within a specific cell volume range, homeostatic range, okay? So here's some of the channels that are open. Remember, these are mechanically gated channels, right? These mechanically gated channels are attached to the cytoskeletal elements. So when the cell starts to swell, it stretches those cytoskeletal elements and opens these channels. In this case, we're talking about, if you guys are looking at this panel, this set of panels on your right-hand side, it's opening up on the top one here, a potassium and chloride channel. Potassium and chloride go out. Or it also activates this potassium chloride co-transporter. You know what symporters and co-transporters are, right? It's using the potassium gradient to also move chloride out. Then you've got a series of exchangers, secondary active transporters. This one right there. My arrow isn't working very well right here. It's the third one down. You've got a sodium-calcium exchanger. That's a secondary active transporter. And a calcium ATPase. It's a primary active transporter that's using ATP, the hydrolysis of ATP, to remove calcium. And then finally on the bottom here, it revs up the pump. Remember, with every turn of the pump, you're removing one osmolyte. Now, what was the take-home message that I told you when we rolled this out? What does water do? If these solutes are being moved out of the cell, what happens to water? Yes? Perfect. Okay, so that's the take-home message. Water is going to follow the solute movement if there's a pathway for water. Water's always going to follow that solute movement if there's a pathway for water. So it makes sense on this left-hand side to swell the cells in the face of a hypertonic solution. You're going to be moving solutes in the opposite direction. You're going to be moving solutes into the cell, and water's going to follow. Okay? Any questions about this? This is called regulatory volume decrease, regulatory volume increase. Okay. All right, so that's really good physiology right there, right? Homeostasis, forces and flows, right? It's one of our three major themes that we talked about on the very first day, Okay, so let's move on. Let's talk briefly about calcium regulation. Calcium is a very important second messenger, as we learned with the GQ coupled receptor. Why it's tightly regulated in neurons and muscle. Again, it's a very important second messenger. So a lot of times, an influx of calcium causes vesicle fusion and neurotransmitter release in neurons. But even more importantly for us, when we learn muscle, intracellular calcium is responsible for muscle contraction. So it's tightly regulated in every cell. The extracellular calcium is usually about 1 millimolar. Intracellular calcium is about 10 micromolar. Okay. All right. So actually, let's see. Is that right? Yeah. Okay. So let me talk about some of the players that help to regulate intracellular calcium. You have the sodium, there's my arrow. You have the sodium calcium exchanger right here. It uses the sodium gradient to remove calcium. It's called the exchanger. It is an antiporter or counter transporter. This is going to be very important with the heart. Okay? It's the way that the heart cells actually remove calcium. Dihydropyridine receptors, we're going to learn about that when we talk about skeletal muscle. And the plasma membrane calcium ATPase, that's a primary active transporter. Now, this is a really nice figure. It actually is really pertaining to muscle in particular. The smooth ER in muscle is called the sarcoplasmic reticulum. Okay? And there's this calcium pump here that resequesters calcium into the smooth ER. This is called the sarcoplasmic and endoplasmic reticulum calcium ATPase, the SERCA pump. And we'll learn about that for exam 2. That's the primary active transporter that transports calcium against its concentration gradient back into the SR. Okay? So again, calcium is tightly regulated in these cells. Any questions so far? All right, so another regulatory mechanism that uses these transporters and channels, right, to maintain homeostasis. All right, finally, this is regulating pH within the cell. All right, this is a little bit more difficult. Let's draw a cell again. Think about what we're talking about here. We have 2 players. We actually have a sodium proton exchanger that we've talked about before. It uses the sodium gradient to remove protons from the cell. Okay? And we have a chloride bicarbonate exchanger. I'll put that on this side. Chloride bicarbonate can go in either direction. Just remember that it is an exchanger. So I'm going to say it's going to use the chloride gradient to remove bicarbonate from the cell. All right, chloride bicarbonate exchanger. All right, so the way to read this graph is, let's talk about this box right here is supposed to be a cell, a typical cell. And inside the cell, you actually have buffers in place. Okay? Those buffers help to maintain a specific pH. If you overwhelm the buffering capacity, you're going to change the pH pretty rapidly. Okay? So pH is a measure of free proton levels. Does that make sense to everyone? Free proton levels, free hydrogen ion levels. Okay, So let's take it from here. We're going to take it from the left-hand side first. Normal pH is about 7.2. Okay. Now, if for some reason the intracellular pH starts to fall, so looking at this, we're going to go down in this direction, right? What's happening is we're accumulating free protons within the cell. Okay, so pH is falling. The lower the pH, the higher the acidity. Okay? What usually happens is it increases, there's a mechanism in place to trigger an increase in the sodium bicarbonate exchanger. Makes sense, right? What it's going to do is it's going to rev up this exchanger. Using more sodium is going to go down its concentration gradient, and it is going to remove protons from the cell to try to maintain homeostasis, pH, within the cell. So it's going to increase the sodium proton exchanger's activity. What that's going to do is it's going to bring the pH back up. So then you go to the right-hand side. It brings the pH back up to normal levels. Okay? All right. So now what happens if the pH rises, right? If the pH rises, what's happening is you're getting a decrease in free proton levels. So what it tends to do is, obviously, it does the opposite with the sodium proton exchanger. It's actually going to inhibit it or slow its activity. You're going to get an accumulation of protons as a result. But it's also going to increase the chloride bicarbonate exchanger. How does that work? How does that help? What do you think about that? The orientation is correct. It's going to use the chloride gradient to remove bicarbonate. What does that do to help bring the pH back down to normal? Take a look at your major buffers within the cell. Bicarbonate is one of your major buffers. The thing to remember is if you remove your buffer, you're going to increase the free proton levels within the cell. By removing your buffer, your major buffer, bicarbonate, you actually help to elevate free proton levels. Does that make sense to everybody? That actually then helps bring the pH back down to normal. Great physiology, right? Again, mechanisms in place to maintain homeostasis, all about forces and flows, moving these electrolytes. Okay? Any questions? Pretty good? All right. I better stop for a minute. Any questions? No. Okay, great. All right, so what we're going to do now is we're going to go on to epithelial cells. I actually want to bring a lot of concepts. We're going to do a little reviewing. I want to bring a lot of concepts together so you can see how it all fits together. Epithelial cells are really, really important. Okay, understanding epithelial cells are going to help you understand lung function, kidney function, small intestines. These are basically the cells that are the barriers between the outside world and the inside of your body. All right? And knowing something about their function is really important. Okay, so before I move on to, I heard that there's actually some issues with the video recordings. Like you can hear the audio up until about 20, 30 minutes and then it shuts off. I don't know if it's something in this room. In 64 Biosci, I'm not having any trouble with that, but maybe there's something with the equipment in this room. So I can do one of two things. There's already a recorded lecture on PowerPoint, right? So you can access that, find the slide that's missing, and you should be able to find the audio with that. Or if that's not working, I think it is, I can bring in the recorded lectures from the first section, which are exactly the same, okay? So let me know which works better for you, and we'll just see if it continues, okay? All right. So let's go ahead and open up epithelial cells. Oh, before I do that, before I get to epithelial cells, I actually want to roll out something that I didn't roll out the other day. And it pertains to your homework assignment. Okay, so everybody should have gotten, our stapler isn't, our copier isn't working, it doesn't staple. So this is just fine anyway. You have the case study for cystic fibrosis right here, it's front and back. And then the questions on the other sheet. Really, we just need the questions. If you could just answer the questions from the case study, this is all we need for problem set number 3. You can just turn this in. Make sure you have your name at the top. So everyone should have the case study, problem set number 3, and the questions that are associated with it. Anybody need a copy or everybody has some? Okay, yeah, I can give this to you. There we go. And if we have some extras, okay, yeah. Are there some extras up there or extras around this way? All right, great, thank you. I do have the questions. Are there the case study around? Anybody need a copy? Okay, yep. Do you all have the case studies on this side? Okay. I'll find a case study for you too. Oh, there's one right here. Okay, sorry about that. There we go. All right, so let's roll this out. This is not going to be due then until next Tuesday. Every section has a full week to do this. This will be due next Tuesday. All right, so let's go ahead and open that up. All right, going back to active transport here. Let's see, oops. I don't think this is it. Okay. Sorry, got these different ones floating around for instructor copy. Okay. All right, well, what I'm gonna do then, it's really some pictures that I have, and I have the Facebook page already kind of set up. So in this case, I want to just feature this little boy here. I have asked my former student if it's okay that I show, his name is Spencer. Spencer is a super cute little boy. I want to put a face to a name.
Spencer, why do you look so mad?
What?
The house is that messy? You do all this work and nobody helps, huh?
He really likes cleaning the house.
It was a bad deal. They wanted to break us up.
That's exactly the point,
Miss Lockhart. You have creditors, you owe them money.
So Spencer,
In your self-fulfillment.
What are you doing?
Spencer is actually a kid who was born with cystic fibrosis. Okay, this was in my slides, but I can show you through her Facebook page. He tends to get cold super easy, so she has to be really careful wearing a mask all the time. And he's a little kid that on almost a daily basis, he has to wear this vest, right? This is a type of vest for kids that have cystic fibrosis. It actually is very jarring. It kind of moves him about like this. It helps to get a lot of the mucus that's stuck in his lungs dislodged so that he can cough it out and get that out. So super cute little kid. There is something that, if I can find it in just a minute, one of the major issues that they saw early on was that, was that he actually, when he sweats, he doesn't have the capability to reabsorb salt from his sweat gland, right? In the duct portion, salts like sodium and chloride actually get reabsorbed. So what happens is when he sweats, he gets these salt deposits on the top of his head or on his forehead. There's a picture, if I can find it right before we leave. My former student actually circled it, and you can see just big sweat or salt deposits on the top of his head because of the disorder, the disease. They diagnosed it when he was like 3 years old. Also, instead of chloride secretion and water secretion in the intestines, what happens is they don't have the capability of that kind of transport, so they get mucus plugs in the small intestines. That's the first thing that they notice when he's born, and he might have an obstruction, and then that's a very good sign of cystic fibrosis. Okay, so again, with my former student's permission, she's very intent on educating people on cystic fibrosis. So she always wants to tell Spencer's story. What I'd like you to do is a similar story, read through what's happening with this particular family and learn a little bit more about cystic fibrosis. We're going to be talking about it today with epithelial cells. So I want you to keep this lecture in mind when you actually go through these questions. Okay? All right, so let's go ahead and talk about epithelial cells. And I'm going to do a little chalk talk in just a minute too after I go through some of these slides. All right, so again, I can't stress enough how important it is to learn about epithelial cells. It's really going to help you with the function of different organs. We're going to broaden it out when we get to the different organ systems here very soon. Let me just give you some information. Here we are. We've learned about signal transduction pathways. Those were our G protein coupled receptors, cell volume regulation, regulation of intracellular calcium, pH regulation, and now epithelial transport. So the main idea with epithelial cells is these cells actually, a lot of them have a high rate of transport. So they help with what's called secretion, moving solute and fluid in one direction into the middle of a cavity, or absorption, like in your small intestines when you absorb calcium or glucose. That goes in the opposite direction. These epithelial cells are very important in that transport. So think about it this way. The epithelial cells are the boundary between the outside world and the inside of your body. So think about your lungs, right? Your air from the outside world goes into your lungs, and it's the epithelial cells that air encounters pretty much first. Okay? These tissues have physiological functions such as respiration, digestion, and ion and water regulation, osmoregulation. All right. So to first start off, there's 3 different types of organization that these epithelial cells exhibit. First one is flat sheets, just like your epidermis. Now your epidermis, those epidermal cells, they really don't have a whole lot of transport properties, but they are a barrier, right, between the outside world and the inside of your body. The next one is tubules. And these are really important, tubules. Let's take a look at it. I'll come back to this slide. The kidney is a perfect example. So take a look at this. These epithelial cells form a tubule, like a cylinder. And once they become contact inhibited like this, they're able to become polarized. I'll talk about that in just a second. What that means is they start to sort different proteins to different sides. So the side that actually faces the lumen, or the interior of the tubule, this is called the luminal side, makes sense. It's also called the apical side, all right? Now, the other side that faces the blood supply is called the serosal side, or the basolateral side. And I'll roll out those formal definitions in just a second. But you can see how you can start to get vectoral or one-way direction of specific electrolytes or fluid, okay, if it's polarized like that. And we'll talk more about that in just a second. But I want you to see how it's organized. So tubules are another organization. When we talk about the nephron in the kidney, it's all about epithelial cells. And again, this is why we started off the semester with diffusion and osmosis, because the nephron is all about diffusion and osmosis. And then glands, okay? Glands, everyone kind of knows what a gland is. There's different complexities of glands. The crypts in the small intestines, these are the simplest. They're kind of like just a U shape, okay? They don't have much distinction in that, and they do a lot of secretion. All right, acinar glands look like number 2 here. And when I was talking about sweat glands with cystic fibrosis, sweat glands are acinar type glands. Okay, so take a look at number 2, right in the middle. That's an acinar type gland. It has a bulb portion. This is called the acinus, and a duct portion, right? And that's where a lot of times the interior fluid is modified. In cystic in sweat gland, it reabsorbs salts, and then it delivers a hypoosmotic solution onto the skin. All right, so salivary and mammary glands are also other examples of acinar type glands. Also, the pancreas is an acinar type gland. Okay, pancreas has acinar type glands. And then finally, the most complex are endometrial glands. These are actually glands that are present in the uterus, females. And number 3 is a good example. It can be very complicated, lots of branching. It can even be coiled. Okay, so very complicated glands. All right, so now that we're talking about glands, I want to give you a couple more terms. We'll be talking about this especially when we get to endocrinology. This is an acinar type gland, as we talked about before. It has an acinus portion and a duct portion. This is a typical exocrine gland, exocrine gland. You can see that right here. Can you see that right? Exocrine. Okay. Because the fluid, the electrolytes, the solutes, the fluid are all delivered to an external environment, like the interior of a lung, the interior of the small intestines. Endocrine glands, they deliver their molecules directly into the blood supply. Okay? Endocrine. They deliver molecules, hormones, directly into the circulatory system to be delivered. Okay? All right, so let's take a look at some interesting features of epithelial cells. A lot of times they grow individually, but they usually form sheets. They usually form structures. So what happens is when they when they actually come into contact with each other, they form these particular junctions. So there's different types of junctions. We're going to be talking about tight junctions a lot. Tight junctions, you can see here, are junctions between the two cells. This is cell A on the left-hand side and cell B on the right-hand side. Once tight junctions form, that signals the cell to become polarized. Starts to sort different proteins to different sides. Okay? Now, other types of junctions that are a little bit more stable are called desmosomes. They're also called spot welds. And you can see here they have these glycoprotein filaments, and then they're attached to the cytoskeleton elements. Okay. Now, here's one that I'm pretty sure is going to be on the exam. Okay. I really want you to know gap junctions. Gap junctions are actually ion channels. All right, so each of the two cells express what's called a hemichannel. And these hemichannels come together to form a complete channel. All right, so these hemichannels are called, they're made up of 6 different connexin proteins, and they come together so that ions can move from cell to cell without having to go into the extracellular environment. They can move from cell to cell. So why is this important? Let's talk about your heart for a minute. One cell depolarizes in your heart, and that depolarization wave moves from cell to cell to cell to cell so that you can get a coordinated heartbeat, right? It's because all of your cardiac muscles are attached together by these gap junctions. Again, it allows for electrolytes to move from cell to cell. Okay? So, formal definitions: gap junctions, specialized protein complexes that create an aqueous pore between 2 adjacent cells. The chemical messengers can travel from the signaling cell to the target cell without ever having to leave an aqueous environment. Or in other words, without ever having to go outside the cell. And it typically involves the movement of ions. Cations, sodium, potassium. All right, everybody pretty good with these concepts so far? All right, so the next one, here's 2 new terms. One is called transcellular. So if a molecule or an ion moves through the cell, this is called the transcellular pathway. Many times ions can move in between epithelial cells too. That's called the paracellular pathway. And you'll get how that works in just a second and why I'm telling you about these terms. All right? So let's take a look at the typical endometrial gland. Remember, I told you it's very complicated. These are some nice slides. You can actually see the epithelial cells right here, cross-section that line the gland. These are the epithelial cells. You can see that it's highly branched, and here's another one that's highly branched and even coiled in nature. So these are some really nice images. What you can do is you can isolate glands. I think this is interesting as well. They are heavier than just individual cells. So you can spin them down. You can, isolate those clumped cells. Those are the glands. You can place them in a tissue culture dish, and the cells will start to grow out radially from the gland until you get something that looks like this on the right-hand side, a monolayer. You can see that a monolayer has formed. These epithelial cells are in contact with each other. They have tight junction formations, and they have started to polarize so that you can study them, which is really cool. Anybody know Dr. O'Grady in the animal science department? This is actually from his laboratory. Okay. All right, so what happens is once the cells become contact, they come into contact with each other, they start to increase their resistance. Let's talk about resistance. This is the difficulty at which ions move across the whole monolayer. Not just in and out of the cells, but the whole monolayer. So as tight junctions form, it starts to restrict movement across the paracellular pathway. And you can see there's an increase in the resistance, which you can measure with an ohm meter, actually. Pretty easy to measure. Okay, some review slides here. Here's our tight junction formation at the top here, these yellow things. Once you have formation of the tight junctions, epithelial cells become polarized. So some transporters are transported to the apical side, right? This is like the lumen of a cavity. And then some other transporters, different transporters, are actually transported to the basolateral side. Okay, so here's the names. Apical side is the luminal side. Basolateral side faces the blood supply. Pretty good so far? All right, now it gets really super complicated. All right, kind of crazy, but I'm going to walk you through it. All right, so just taking a look at this particular picture here, you know a few things already. What G protein coupled receptor is this? UTP is binding to a purinergic receptor. What's happening? It's GQ. It's actually listed right there. No, it's not. The G alpha, this is the GQ pathway. All right, this is actually in a journal, okay? And what you can see here is the GQ pathway is opening up a couple of chloride channels. All right, so let's think about that. Let's think about this. You already have the background to understand this. So, first, number one, you can see my favorite co-transporter. Talked about this before. The sodium potassium 2-chloride co-transporter. It is moving an enormous amount of ions into the cell. This is a secondary active co-transporter. It's using the sodium gradient to bring in chloride into the cell. Step number one. It's the loading step. All right, so now I'm going to have you do something. Everyone's kind of looking at me like, let's say, here's our cell. The outside is about 150 millimolar. Now, because of our loading step, the inside of the cell is going to start to accumulate chloride, right? So let's say it, in some of our problem sets, it was down to about 30. Let's say now it is 80 millimolar. Can someone tell me what the equilibrium potential for chloride is if the temperature is 37 degrees Celsius? Bringing a lot of things together here. Here's our concentration gradient, right? So what did you get for the equilibrium potential for chloride? Anybody need do you want me to put the equation on the board? Would that be helpful? Okay. Oops. Oh yeah, I can erase this one. All right, so the equation, this is how you would set it up. Equilibrium potential for chloride. R is equal to 8.3 joules per mole Kelvin. T is in Kelvin. How do we convert that to Kelvin? It's 37 degrees Celsius, 273. That would be 310 Kelvin, right? Over Z, I'm not going to tell you what Z is, times 96,500 coulombs per mole. Okay? Then we want to do the natural log concentration gradient is built right in. 150 millimolar over 80. Oops, 80 millimolar. That's how I would set it up. Just some extra practice. Anybody got the answer?
I got negative 17 millivolts.
Negative 17? That's right. So, negative 17 millivolts. It's pretty depolarized, right? More positive than you would have guessed. When we were doing chloride before, it was around minus 60 millivolts. So, loading chloride into the cell actually helped to depolarize the equilibrium potential. However, there's still a very high conductance for potassium. If there's a high conductance for potassium, where's the resting membrane potential going to be? Let's just say the equilibrium potential for potassium is minus 80 millivolts, and it's a 90% still conductance for potassium. Where would you expect it to be? Pretty hyperpolarized, right? I'll just tell you, resting membrane potential is still about minus 70. But you know how to calculate it. If you had to predict the resting membrane potential, you would use the chord conductance equation, right? A little bit of a review. That's this equation right here. The conductance for sodium over the total conductance times the equilibrium potential for sodium plus conductance for potassium over the total conductance plus chloride. It's all the ions moving across the membrane. We'd have to put conductance for chloride over the total conductance times the equilibrium potential for chloride. All the ions moving across the membrane, you can predict the membrane voltage. All right, so taking a look at this, you can draw a graph. Equilibrium potential for chloride, minus 17. Resting membrane potential, minus 70. So when I trigger the G protein, the GQ coupled protein receptor, and you have this cascade of events that releases calcium into the intracellular space and opens up this calcium activated chloride channel, which way is chloride going to move? Which way is chloride going to go? Here's my reference. I can see some people mouthing the answer already. Yeah, out, right? Put a little negative sign because the resting membrane potential is much more negative. Chloride is going to go out of the cell. Right? Good job. All right, so now let's think about it this way. You have a one-way direction. I'm going to say vectoral movement. That means it's going in one way. You have a vectoral movement of chloride through the transcellular pathway, right? It's going from the basolateral side into the interior of the gland, right? Everybody with me on that? All right, so now here's a little, everybody pretty good with this? I'm going to go ahead and erase it. Now we're going to make it, we're going to zoom out and think about this. Here's a typical gland. Right? Gland. Here's our epithelial cells that line it. See where I'm going with all this? This is a monolayer. These are the epithelial cells. So how does electrolytes and nutrients and fluid move from a gland, like a sweat gland, into the external environment? Is it magic? not magic. Okay? So what we've shown right now is that chloride is going to move through the transcellular pathway into the interior of the gland. Okay? I'm going to turn to this next one. Well, almost. All right. So take a look at what happens to the voltage across the entire monolayer now. All right? Take a look at this. Here's the voltage. Here it is right here. See this V on the very left-hand side? Because we have this movement of chloride that's moving in a one-way direction, right? It's setting up a voltage gradient across the entire monolayer so that negative is on the interior and positive is on the other side, right? You've now set up a gradient, a voltage gradient, across the entire monolayer, all right? So, I'm going to do that up. Here's the voltage, negative on this side, positive on this side, right? What that does is it sets up a driving force for the movement of sodium across the paracellular pathway. Think about that. Now that you've set up the voltage gradient, it's going to attract sodium, and sodium is going to move across the paracellular pathway in between the cells. Does that make sense to everyone? So now you have this movement of salt into the interior of the gland. What's going to happen to the water? It's going to follow the solute movement. So now water actually starts to move in here. You can see how water and solutes are now going to be delivered. It's just biophysics, right? This is physics. It's not magic. All right, so going to this next one, this actually shows you what's really cool about this. It shows you chloride is moving across the transcellular pathway, sodium across the paracellular pathway. That sets up an osmotic driving force for the movement of water to follow, right? Then what happens? That gland starts to stretch. And what it does is it actually releases more UTP into the interior. You get more UTP, which activates more GQ, which forces more chloride, which forces more sodium, more water, and it's like a positive feedback, right? Until you just start to spew water and electrolytes and nutrients and other things of the like. It's pretty cool, right? That's how glands work. That's epithelial cells. So you can see here, the more water and electrolytes, the more it stretches, the more UTP is released, the more it activates more GQ-coupled receptors. Okay? All right, pretty cool. That's how epithelial cells work. So I want you to think about that. When it comes to cystic fibrosis, yeah?
Can you just remind me what UTP is?
Yes, so UTP is the ligand. That's a really good question. Let's go back. It's the ligand that binds to the GQ-coupled receptor, which then activates more chloride channels to open, vectoral movement of chloride across the transcellular pathway. So it just keeps initiating more cells to secrete chloride, sodium, water. Okay? Okay, so what happens? Let's go back to this one. Here is CFTR. CFTR. If this channel doesn't get trafficked, it usually goes to the apical membrane. If it's not there, you inhibit chloride secretion. You inhibit sodium secretion. You inhibit fluid secretion, right? You don't get fluid in those ducts. What tends to happen is there's a hyper absorption and you tend to get a mucus plug instead of a nice secretion of water and electrolytes. So think about that. CFTR is expressed in lung cells, in intestines, in pancreatic duct, in the uterus. That's why infertility is a problem with cystic fibrosis. it is really ubiquitous in a lot of different cell types. So now, think about these transport mechanisms when you're actually doing your study here, when you're doing your problem set. Okay, pretty cool. I mean, it's cool to think about how this works and what happens when things go wrong. All right? All right, so we still have 15 minutes. I'm going to start with the nervous system today. Okay? Got about 15 minutes. We're just going to talk about the nervous system in general. On Thursday and next Tuesday, we're going to concentrate on neurons and action potentials. Okay? So let's go ahead and open up the nervous system. This is very general. If you are in the lab, a lot of our students in our department are in the laboratory. You've probably seen this already. It's good to review, but obviously, for some of you, you haven't seen this material before. You might have had it in other classes, but it's good to go through it. So let's talk about the two major divisions of the nervous system. We'll start off with that. The central nervous system is referring to the brain and the spinal cord, okay? Especially if you have already had neuroscience or neurobiology, this is going to be really review for you. All right, the central nervous system is pertaining to the brain and the spinal cord. The peripheral nervous system is comprised of the nerves that connect the brain and the spinal cord to other parts of your body, like muscle, glands, and sense organs. And the neuron is the basic cell type of both systems. These are the signaling cells. We're going to talk about glial cells in just a little bit. Those are more supportive cells. It's the neurons that are really transmitting those signals, those electrical signals. Okay, so let's talk about these two different divisions. Okay, we have the afferent division and the efferent division. Afferent is at the top there with an A. Efferent is at the bottom. So we're going to be talking about afferent arterioles too when we get to the circulatory system, and efferent. Whenever you see those two terms, think about this. Afferent means you're transporting something to, in this case, to the central nervous system. Efferent is actually transporting something away from the central nervous system. Okay, so the afferent division is basically transporting a signal to the central nervous system. Efferent are signals away from the central nervous system. Okay, so the general somatic sensory system, you can think about that as these are sensory neurons that basically transmit pain signals, temperature signals from different parts of your body. The general visceral sensory system is actually transmitting like pain and uncomfortable kind of feelings from glands, blood vessels, and basically your internal organs. Visceral is referring to internal structures. All right. And then the special sensory system is basically some of your organs that transmit sight, hearing, things like that. Olfactory. These are your senses. All right, so the efferent division at the bottom there, let's start off with the somatic motor neurons. So on Thursday, we're going to specifically zero in on a motor neuron, and we're going to learn about graded and action potentials. Okay? So this is part of the efferent division. And this is both of these are in the peripheral nervous system, the afferent and efferent. And the somatic motor neurons are those neurons that control muscle. They're voluntary. You control those the activation of those neurons and contraction of muscle. Okay. And then the autonomic nervous system, right below there, is the involuntary arm of the efferent division. So you don't control this. You can try to control it a little bit, maybe with breathing rate, but that is your sympathetic, parasympathetic, and enteric system, nervous system. So the sympathetic is your fight or flight response, right? Sometimes you feel stress or anxiety, scared, right? That's all part of the sympathetic nervous system, your fight or flight response. The parasympathetic is the opposite. It's the resting and digesting arm. And it actually has the opposite effects in a lot of organs. Sympathetic causes your heart to race. Parasympathetic slows it down. Both of those are involuntary. And then the enteric system, maybe you didn't know this, but your gut, small intestines, large intestines, are one of the most innervated organs in your entire body. You have more nerves to that part of your body than almost any other organ. Enteric nervous system. All right, so again, we're still concentrating on the peripheral nervous system. You can see the afferent neurons at the top here are actually transporting signals from different parts of your body to the central nervous system. Interneurons are neurons within your central nervous system, your brain, that are altering that signal. Now, think about this. 99% of your neurons are actually interneurons. Only 1% make up the peripheral nervous system, the afferent and efferent. So that's pretty amazing. All right, and the efferent neuron, like a motor neuron, transports signals from the central nervous system to, say, muscle glandular neurons. Now, if you need some text associated with that, you can read through this at home. This will help you kind of remember what I just said. And let's now talk about the difference between neurons and then glial cells. So the neurons are what's called the nerve cells. And they are amitotic, meaning they don't divide. So that's why spinal cord injuries are actually really devastating, because nerve cells don't really regenerate, they don't divide. Now they also have a very high metabolic rate. There were a few studies a few years ago pertaining to Alzheimer's disease, where they said that this may be a diabetes type 3. So Alzheimer's disease, think about that. They were thinking because these nerves are so highly, they have such a high metabolic rate that basically they weren't getting a lot of glucose into that area, and that it might be a diabetes type 3 that was causing Alzheimer's. It's kind of still being kicked around, but one of the treatments was maybe glucose nasal spray or insulin to try to help these neurons absorb more glucose. All right, and then clusters of cell bodies in the central nervous system are called nuclei. And just remember that neurons are not the most abundant in the central nervous cells, but glial cells are. Glials are supportive cells. And you can see here there's a lot of communication, especially with interneurons, where one neuron basically sends a signal, releases neurotransmitter, and signals another neuron. So a couple of new terms. This orange cell at the top, you can think about it as a presynaptic neuron to the green one, which is the postsynaptic. Okay, so this one right here, a single neuron, postsynaptic to one cell, can be presynaptic to the other. Does that make sense? So if this axon is signaling the orange one, you would actually call this the presynaptic and this the postsynaptic. Now let's take a look at the orange and green. Now the orange one actually signals the green one. This becomes the presynaptic. This becomes the postsynaptic, okay? All right, so there's a lot of communication. We still have a few minutes here. Let me just roll out this particular scientist. Like I said, I always like to kind of go back and talk about scientists that really made a huge impact. This is Santiago Cajal. Maybe you've heard of his name before. He was very instrumental in actually characterizing neurons. Okay, so let me just show you. This is a drawing. He also did a lot of work looking at retinal neurons as well. And I'm just going to show you this particular video. I think this is interesting. I've seen other videos about him as a kid. He was actually quite the juvenile delinquent. He was always in trouble. He was always getting kicked out of school. He's actually got a very fascinating story. He lived like right at the turn of the century, 1900, 1920. And also, this is a good story. Golgi did a lot of work with neurons as well. And I guess they both won the Nobel Prize in the early 1900s. And neither one of them were in agreement. So a lot of drama happened during their speeches at the Nobel Prize, which was really interesting too. So let me just show you this particular video.
Just want to talk about this image that Cajal drew of the retina of the lizard. Cajal was very excited about the retina, I think in part because of his connection to it as an artist. And what's really interesting about this image is that he's depicted many different types of retinal ganglion cells. And in particular, there are many different types, and we know now that the reason for these different types is to do with the function of these different cells. At the time, Cajal would not have known what the functions were because he was not able to image live tissue. But he was able to notice that these things did have different types and therefore could postulate that maybe the functions would be different. And I think this is really a testament to what Cajal was able to infer with the limited data that he had, as well as how we were able to then use that information with new techniques in being able to do imaging within live animals to determine the really interesting and varied functions of these different types of cells. So here you have cells that might correspond to, for example, seeing a red or a green object in the world, or seeing a moving object or a still object. And now we know a lot more about the retina because of these new functional techniques.
All right, MIT Museum. Okay, that's it for today, everybody. I will continue on with the nervous system, and we're going to dive right into neurons on Thursday. Have a good Wednesday, everyone. And if you have any questions, just come on up. All right. Yes, and the questions are great. Yeah.
Are we good for Thursday?
Yes, sounds great.
All
Thank you,
right.
Kara.
I'm
Yes.
gonna go find my wallet. I can't find it the whole time. I'm like on the verge of crying.
Oh, I'm so sorry. That is stressful.
I put it somewhere. I
You
don't
know,
where.
one year, this is many years ago, I thought I was going to be cool and have like a wallet that guys carry, and so I always put it in my back pocket.
Yeah.
And I was walking across campus over by Scotty's house, and it kind of works its own way out. Some nice person actually called me.
I really hope so. I've got like my student ID in there too.
I'm so sorry. I hope you find it. Hi.
I won't be in class this Thursday because
Yes.
I'm going to dairy
The
judging.
dairy judging. Yep. No worries. Thank you for being my volunteer.
Yeah, no problem. I just thought about talking to you about that.
Yeah, no
So-
worries. So I will, like always, I will hopefully capture the entire
Okay.
recording.